Hi everyone. In this video, we will explore another tool for single cell annotation and it's AI based. So that is mLLMCellType. So basically it's an open source, scRNA sequencing cell type annotation tool using multiple AI models with consensus based cell type identification. Okay. So this is a paper. This is not yet published. This is on the, bioRxiv, journal, but it's a very interesting concept. So what they are doing, so you can choose multiple LLM models. So you have different provider, a GPT, Claude, Gemini, Quen, any provider you can choose. And so then you can provide the same query against all these multiple LLM models and then come to a consensus, and then based on that you will get that answer. So that's how it is. So basically this is a unified interface that allows you to you choose any LLM model, and based on the consensus it will give you the results. Okay, so and the URL is mLLMCellType.com. Okay, which let's go over here. So this is the paper that I was talking about. Okay, and yeah, this is the website. You will see mLLMCellType.com, and yeah, so you can do directly over here. And yeah, also they provide the GitHub repository, and you can programmatically interact with that. Okay, so let me come here and just tell like what it does. As I said, it integrates 6 state-of-the-art models. In the paper, according to the paper, it is done, but now the interface provides you flexibility to choose many LLM models, okay, from different providers. And then the beauty is like, this is very important part, the iterative deliberation. Means what internally it does, like it asks the same query to individual model and let them respond, and then if they disagree, right, so then this model will talk to each other, and they share their reasoning, like why this model predicted that way, the other model predicted. So they will talk to each other. Okay, so there is a orchestrator is there in the middle, which will gather the output from each of these one, and based on that it will try to orchestrate the talking between these each model. Ultimately a consensus checker, usually the powerful model like Cloud, Sonnet probably you can choose, which can confirm that, hey, okay, they have reached the threshold, and then this is the final output based on the reasoning like that. Okay, this way this gives you a confidence, like multiple LLM model have multiple intelligence, so you can leverage those to provide the get the accurate output. And input is simply you need just the gene list, marker genes list, because this is all LLM, so they don't understand expression, they just need the marker gene list. Okay, and yeah, I think they provide some hierarchical strategy, like if a big cluster is there, then you can subdivide into multiple subtypes as well. Okay, so what's the advantage, like collective intelligence. That means the consensus process actively suppresses individual model hallucinations. For example, if you are using just one model, you may get a very probability that you may get those model can hallucinate. But since multiple model are telling the same thing, that means that gives you more confidence, right? And transparencies. So unlike black box algorithm, like certain LLM, but it provides reasoning chains, right? So it will also tell why the particular cell type is assigned in the given type, so which will help us to understand further, investigate or interpret the results what we get from these LLM models. And yeah, uncertainty means like uses consensus proportion and Shannon entropy to flag ambiguous cluster that actually need a human expert's eye. This is another features that they provide, like something that needs very less confident by LLM, so that you can go with a human in the loop kind of thing, so you can review and you can proceed for that. And the only thing, like again, there's for the paper it is much cheaper it says, but yeah, if you have to put some money to do it, because because you have to use these models, right? But to try out you can use the free model, but actually in real use case you'll have to pay some money to all these LLM models. Okay, so let's come over here. So I'll just first walk you through the UI part, how does it look like, then we will go to the program part. Okay, so you can simply say get started. Okay, and so you need to upload your marker genes, and the genes, it will look like more into like gene by sample format. Okay, so I have let me load the data and sample data. So yeah, so the data will looks like this in this example data what I downloaded. So for example, you have cluster 1 to 3, and these are the genes cluster 1 marker, these are the cluster 2, these are the 3 markers like that. Okay, so now if I go back here, so you can choose what species you want. Okay, and let's say this is a human data, and I can choose, let's say, blood, and maybe yeah, let's say blood. Okay, I'm checking, searching for immune, maybe let's say it's a blood. Okay, human. And then that's the important part. Here you have to import the API keys. Okay, so let me pause my video, otherwise where is the I'm just thinking. Okay, so I think you have to import the API keys. Okay, so let's do one thing. We'll upload a env file here. Okay, I'll show you how to do that. But here you can see that there are different LLM models are there. OpenAI, these are the models you can choose, and but again you have to provide the API key over here. Okay, and yeah, Anthropic means Claude, Anthropic, like that. But and there's something called Open Router is there. Open Router is nothing but it's a unified interface. Okay, where you can go, for example, this is the Open Router. Okay, so you can just create an account and you can pay some credit, and then you can be able to play with n number of model. Okay, so Open Router is nothing but a kind of interface of all other LLM provider. So instead of 10 API keys paying separately, you pay to Open Router, and Open Router will take care of that. So that's the behind logic behind that. Okay, so I have already created an account here. But if you go to models, right, and just say text model and pricing, right, you can just simply drag down to free. Okay, so there are, I think, 27 free models are there, which we can use to play around, this just to show you. Okay, but some I have tried few, it did not work. Some models work, some doesn't work, but yeah, so that's the thing. If just for trial purpose you can do that. So what we can do, like, for Open Router also, you can just search for free. So you see these are the free models, wherever free is there, that we can use. Let's say, for example, at least minimum 2 models you need, because this is a consensus it needs to do, right? So maybe let's do these 2 models, and then we'll first to upload the API key. Okay, so let me pause my video and I'll get back. Okay, so just to we need to upload the environmental file, right? So the environmental file will look like this. So here you can specify different keys. So OpenAI, Anthropic, Gemini, DeepSeek, Grok, Qwen, like that. So instead, what I have done, I have created a file where I have uploaded just kept only these 2 line, this single line, Open Router API key equal to the API key mine, and I saved that file, and then I uploaded here. Okay, once I uploaded it, and then I just did a testing over here. So test. So I've already uploaded. So once you test it, right, it will verify that, it will say that it is all verified. Okay, request timed out. Okay, let's check again. Okay, sometimes it API hits probably. So okay, so now you see my Open Router API key successful. So now I'm good to go with using these 2 model now. Okay, so DeepSeek chart, and these are free, because that's what I think. Okay, and yeah, so this is my input data. I have uploaded it like gene by this, means for every cluster what are the genes, and you can choose it, and then you just simply and some other parameter you can use, consensus check model. Here also you can use any of these 2. So by default, you can select, let's say I want Open Router, the 3rd model I can choose as well. Okay, but let's do that. Okay, so you can do the start analysis. So now we have using these 2 free model, and it is now querying against that. So it will take some time, but meanwhile, if you come over here, Open Router, what will happen? So you can go over here, Open Router, and on the left hand side you will see the activity. Okay, so this is what so far I'm trialing. It is it will consume some money. Okay, so for example, I was trying DeepSeek R1, which was not it was not free. So for this example, it consumed me different for every cluster it consumed me this money dollar amount. Okay, so it's very negligible, but yeah, now okay, but the real data it may increase. So but now you see it is, I think, doing some query over here. Is it started? Okay, yeah, we got the results. So like for example, 2 models it used, and this less time it took. I don't know whether it took some results or not. Download the results, permission denied. I don't know. Download the logs probably. Yeah, what does it say? So yeah, so it did something, but yeah, I think it did not work. Okay, that's interesting. Yep. Download discussion. Let's say whatever that is discuss, right? We can even download those discussions. So let's see. Yeah, I think this nothing has happened. Okay, sorry for that. I initially I tried, it gave me some results, but now it is not giving. So that's why these are very non-deterministic, right? Okay, but you get an idea, right? Because when we use actual model, you will get some results. Very easy interface. You upload the data, just choose what LLM you want, and uh, submit it. So it's all about the money. So if you have money, it will work, and you have to pay some money for actual work. But it's very smooth, you see. And if you read the paper, they have done, that's the thing, right? So they have used multiple different this thing, and finally the initially round one they do the analysis and discussion, and the round two they further refine, and they do the evidence review. And so that way it is much more better than one LLM telling. Instead of that, multiple LLM should come to a consensus, right? So that's a good thing about this paper. Okay, so let me quickly switch to how to do on programmatically. Okay, because that gives you more flexibility actually, rather than relying on the UI. So they have both R and Python based. Okay, and so that's what they have seen. It includes all the LLM models. And yep, so if you are using R, so you can simply say mCellType installation, and then Python for you have to use pip install. Okay, and then you can also choose just particularly which particular model, but that's not needed. And but only thing, like, you have to use the keys, right? So I will just explain the R1. In Python you can use this and just do the same thing, but I will just explain the R1. Okay, so let me go and check here. Okay, so this is what how you install it. Okay, and just set a working directory, and this is what you can set an environmental variable. Though I'm just writing open router key, that is some I've just hidden it. So but you can create this variable, and so now you can load the all libraries. Okay, and initially this is the PBMC library, PBMC object, which will be 0 like that. And you can store it because this is downloading something, right? It will download reasoning and everything. So instead of every time going back, it stores in this current directory the log and everything, whatever the information it stores here. Okay, so that's what we are creating a cache directory here. And for now, the top markers means like from the Surat pipeline. I'm just for now, because I just want to demonstrate you, because otherwise if I show you for all the cluster, right, 10 cluster, I will consume more tokens, right? So what I have done here, just from the top marker for the first cluster, and just 20 markers I'm taking. Okay, so if you see the C1 marker, it will look like this, right? So these are my marker genes. Okay, now we will take these marker genes and we will query against this one. Okay, so there is a function called interactive consensus annotation, and here you pass that marker, and you can give some tissue name, because that will be the context. So you can say here blood, lungs, you can write these, all will go to the LLM as a context, and here you can pass the model. Okay, so what I did here, I went over here and I went to model here in the open router, and here I choose 0, and yeah, text I selected, and when I selected one model, and and something I tried, but only one working. Okay, as of now, the other one is somehow it is not working. This OpenAI GPT OSS 20, it did not work for me. Okay, so yes, so you can model name, you can do like this. For example, if you select this name, right, so when you just copy this one, so that model name will get copied, and you can just pass what all models you need. So minimum 2 models is needed, more is the better. Okay, but again, you will have to pay more to multiple LLM model. Okay, so for example, here let me try this one and this one, and of which only this is the one working, this one is not working. But just to make it work, right, this function, otherwise it will fail that minimum 2 is needed. Okay, and here I'm passing my OpenAI key, and which I have set this variable. Okay, and just you say that how many genes you want to take, and some controversy threshold and entropy threshold, those you can do it. Okay, so when you do the right, what it will happen, it will, now you see it is now starting like some timestamp. It creates a session ID, and then it tries to phase one it is doing. So what is doing, getting initial prediction from all the models, and total two models. One is this free model Quen, and the another Quen, the free model. So now it is submitting to these two LLMs via open router, and yeah, something it is happening, so we'll have to wait. So meanwhile, I can come back here and I can check the what all the usage in the open router. So you can see that this is April 1, time is 12:49, and you can see this, this is submitting, and now you can see around it is 98 to 1610 token, that many tokens were consuming. Okay, and ideally it is free, that's why it is not needed, but here you can see equivalent. The other one, for example, for $800, this many tokens we consume. Okay, so yep, so you can further click what is happening here. View raw data, what all queries it went. Okay, but let it come back here. So now what it did, see, it gave me an answer like, so it is doing it says that phase 2, first phase it did identification, but here the some API did not work. The second one, as I said, right, only one is working. And the second phase, what it did, like identify controversial clusters. Okay, and for that, then analyzing cluster 0, the first one. And so because only one we got right, so that's why we got a response, not enough responses to check consensus. Okay, and something is happening, cache result, something. Finally it will say that the cluster 0, which, and as per the initial prediction, this was naive T cell, and so it will say that all the model in that discussion log predicted this naive cell, but the final consensus is this one. Okay, so something, it will give you some message, and it will say some warning that I could not get from this one. Okay, but the results will be stored in this consensus result, right? So you can say that how many sorry, this one, so what all variables we got, like the initial results, like that. So we can simply do view consensus results. So you can see that this is the initial results. Okay, so initial result, individual prediction, it says that it is naive T cell, and there is no controversial results. Okay, and final annotation is, it is giving something. Okay, so this is the initial result, but this is the final annotation, is the naive central memory T cell. This is the final it is giving me as a recommendation. But this is the interesting part that I like, the discussion logs. Okay, so here this is, for example, it says I think discuss. Okay, let me show from here. Okay, I think that will be better. So we got this variable, right? So you can say that these are the initial results. Okay, so different phases, like initial prediction, naive T cell, then consensus did not work. Okay, consensus did not work, everything. Finally, these are no results here. Okay, and the final annotation is this one. Okay, but this one is the important one, discussion logs. So what all discussion happened? Okay, this is the main information. So we got the response, cell type, this one, and grounds, on what grounds it is telling. So it says that in the cluster expresses these three markers alongside a distinct set of genes associated with lymphoid tissue homing, and this, and so additional genes include other things which are frequently co-expressed in human, this one. Okay, so this is the warrant, is this one, and what is the backing behind this. Okay, and the qualifier and the rebuttal. So this is the way, that's a judgment I told you, right? So there is something called consensus checker, is right, so which will do all these things, like like what is the ground, what is the warrant, and what is the backing, and what is the qualifier, and finally the rebuttal. So this kind of they the legal battle happens between these LLM, and finally we get the conclusion. So that's the way you will be able to use. Okay, but see, the implementation is very simple. Just this is one simple function. Okay, you choose the model you want and set the API key, and rest everything will be taken care of by AI. Okay, and if you don't want this one, because then why to program, right? So you can come over here and directly upload your data, and it will take care of the rest thing. But ultimately, so main thing is like, see, it's like very easy to do when we are doing experiment, when actually when we do, right? So it may consume many tokens, and also that's what we need to put more checks, and instead of relying on such tools, you yourself can build agentic tools, like, so that you can control how the input, how much output you want. You can restrict the token, you can put some guardrails, you can play with the system prompt, lot many things you can do. But if you're very new to this programming agentic, and so then these tools are definitely a game changer. Okay, and once again, as I said, like, don't rely completely on this LLM, because these are whatever they are telling suggestion, they are telling based on the knowledge they have. Okay, but they simply use the marker genes names. This is more of marker based annotation, but it doesn't do the reference based. Okay, so that's what I would suggest. Like, if you have when you are doing annotation, you should combine all these three methods, like do some marker based annotation, whatever the popular tools are available, and then, because these LLM, they will not use those tools, and those tools are scientific tools people have developed, and it is published, well published tool, right? So simply don't assume that AI has come means, those tools are invalid. No, so those tools are very important tools. You use them, get an output, then try the reference based to get an output, then use LLM will you more assist, it will help you to, like, give some scientific justice, scientific, knowledge behind those things. But ultimately, as a researcher, you should combine all these three to come to an conclusion, and do a best case, you know that's what, is all LLM. But it's a very nice, I would say, uh, move towards, interacting so many LLM models, and come to an consensus. Okay, so with that, yep, I think, we covered, like, at least two tools I covered, CellLlama and then Cassia. This is a very recent paper, Cassia. Okay, that I will try to, explain in the next video. So see, this is, I think, 2025 they published in Nature, and this is also very similar concept what this mLLM does. This is also multi-agent kind of thing that I will try to see, if it is possible to do it. Okay, and yeah, some it also, allows to do local LLM. Yeah, this, if they would have given this one, right, local LLM, then it would have been, game changer. Okay, so with this facts, but the same thing what they have done, right, you can do it yourself. Local LLM you can use, like Olama, but different models, and ask the different models to give you the answer, and you can play around it. So lot many scopes are there, and if you are very new to AI and how to use this model, I have created some other videos around it. Just do watch the playlist, just search for it, and you may encounter, and you may get some idea like how to build this agentic AI system, and yeah, you can do a lot of things in AI. Okay, with that I will stop here, and next we will see, we'll try to go to Cassia, the last tool that I might incorporate. I hope this video was useful, and it gave you some idea about like how these multiple LLM tools work. And please do provide feedback, so that will help me to understand, like, how good the content is, or if I'm lacking somewhere, and maybe I can provide some better insight and better content. Okay, so thanks for supporting, and thanks for watching, and I will see you in the next video.